Hello, my name is Erica Sung, and I'm a first-year bioinformatics master's student at North Carolina State University. Today, we are going to be talking about protein synthesis and ribosome recycling. To begin, I want to give a quick refresher on the first three steps of protein synthesis, which you may also know as the process of translation. This consists of four steps: initiation, elongation, termination, and ribosome recycling before it starts all over again. Protein synthesis is initiated when a small ribosomal subunit on the bottom binds with mRNA, and then the initiator formal methionine transfer RNA, mtRNA, carrying the fmet binds to what is called the start codon of the mRNA sequence, and the methionine encoding AUG. However, this process isn't self-sufficient, as it depends on the help of initiation factors to start by helping the key components we mentioned, the mRNA, tRNA, and small ribosomal subunit, bind each other. Finally, a large ribosomal subunit binds to complete the initiation complex. As part of the large ribosomal subunit, there are three sites, known as E, P, and A, respectively, from left to right. So the first methionine carry tRNA begins in the middle at the P site during initiation. The next step is elongation, where the ribosome will move on from the start codon onto the next triplet of nucleotides. So when an amino acid is added to the A site, also known as the landing site, a peptide bond is formed by transferring the methionine from the first tRNA onto the amino acid of the second to form the start of a peptide chain. The complex moves down the mRNA, thus moving the exactly one codon over to allow the previous one to exit in the E site and reopening the A site. The elongation stage consists of adding and elongating the chain of amino acids linked by peptide bonds until it is all red and then terminated. Termination occurs by one of three stop codons, UAA, UAG, or UGA, when it binds to the mRNA, leading us right into ribosome recycling. This process involves the recycling of the post-termination complexes. There are three release factors that we should know about to help with the process: RF1, RF2, and RF3. RF1 and RF2 recognizes the stop codons mentioned earlier, but the purpose of RF3 is to free up RF1 and RF2 from the post-termination complex on the ribosome. When the stop codon is reached, then a release factor, either RF1 or RF2, catalyzes the transfer of the peptide from the P site to the molecule of water in the A site, and the peptide is hydrolyzed and dissociates. The last step is the release of the ribosome from mRNA, which is catalyzed by the joint activity of EFG, a translocation factor, and a ribosome recycling factor, RRF. Thus, the idea of recycling is that the ribosomal subunits are split up and available to bind to another mRNA to start the whole protein synthesis process over again.